Thank you for joining again as we go through our story of the immune system and how it is engineered. If you recall, we've gotten now to the point where we've worked through inflammation. We've worked into the bridge between inflammation and the adaptive immune response with the antigen presentation from cells like mononuclear phagocytes and dendritic cells. And now we're ready to talk about the signaling downstream from the general of the immunologic army, who I told you was the T helper cell, down the side of the immune response where cell-mediated immunity will become activated. And if you recall, cell-mediated immunity is designed to identify altered self cells or infected cells. It's the way to tell the difference that is apparently minute between a cell of your own body that has an alien living inside it or has a malignant process going on and a normal cell of your body. It's a very highly tuned and discriminating mechanism of immunity. So realize where we left off before was the discussion of the fact that if you think about that APC detecting differences in the inflammatory site that came from things that were intracellular and were presented in the context of intracellular cytoplasmic TLRs like TLR3, 7, 8, and 9, then realize that the innate lymphoid cells in the area of that original damage are going to signal downstream and are going to tell that APC how to communicate to the T helper cell where the process has happened. So as we look at our next diagram, you'll notice, of course, that it is the APC's recognition of those TLRs that were cytoplasmic and the signaling of interferon gamma from innate cells like natural killers that told that APC that what had happened that had caused cellular damage was indeed something living inside that cell. So when that APC ingested the debris of that material, it sent the signal downstream to the T helper 0, who was naive, and instructed him that this was the way that the damage to the system had occurred. And this is done through signaling of cytokines as always. I'm going to suggest to you that you work on getting one cytokine committed to memory at first, and then as time goes on and you have more time, you can go back and work on others. But the one that I think is most important for the purpose right now is interleukin 12. That is a very clear signal from this classically activated APC that tells the mature naive T helper that it should transform itself into a T helper 1. Now, when I allude to nirvana in this diagram, this is sort of a silly thing that has occurred over the many years I've been teaching, that I get to the place where I have the entire immune response on one page, and you're going to see that for the first time at the end of this chapter. But my context here was to remind you that there is a plane of intellectual ecstasy that can be achieved when one has the entire system under control. And we've been building very stepwise and incrementally toward that point. But that's my allusion to nirvana. It is that plane of ecstasy in the Hindu thought process where one does not have to worry about reincarnation anymore because you have achieved the ultimate plane of intellectual achievement. So we're going to get there for the first time at the end of this chapter. Now, if you recall, the T helper 1 cell is going to give signals downstream to the effectors on this side of the diagram. And it's true that when I activate this response appropriately, hopefully I will remove that pathogen, whatever it was, or that neoplastic cell, or whatever it was that was causing the problems. But you have to keep in mind that the immune response is truly a sliding scale of responses. And when I have too much of this type of immune response, the outcome in the patient is type 4 hypersensitivities. Just enough is perfect and I resolve the problem. Too much and I end up damaging my own tissues, as we'll speak about in a later discussion. Now, the early signaling for CMIs, as we said, is the recognition that whatever caused the problem was indeed an intracellular thing that caused the destruction of the cell. And so I have a number of processes that turn this on, which are recognized by TLRs 3 and 7 and 8 and 9, indicating that there is an alternate lifestyle living inside the cell. So when that material is phagocytized after the cell dies, and I have natural cells making interferon gamma, resident natural killer cells, then that induces the APC to signal downstream with those cytokines I spoke about before. And if you'll notice, as we look at the signaling toward the effector cells, it's true that the APC on this side is always the same cell, and the helper on this side of nirvana is always the same cell. But from there on, I have very specific cells that are turned on in order to achieve the removal of very specific pathogens. Now, this system has evolved over evolutionary time to be the battle point between various microbes and our bodies. So realize that as various microbes achieved the ability to avoid one of these responses, then your immune response answered with another one. And so this has been an ongoing process. I think the easiest way to think about these is to notice that the cytotoxic cell, or CTL if you prefer, is the classic cell that will remove cells that are infected with an alien, okay, a virus living inside the cell. The cytotoxic cell is your best cell because it has memory and it will recall what it did yesterday, and it will constantly improve the way it acts tomorrow. The NK cell has evolved to take care of those things in which the cytotoxic T cell cannot take care of. And I'll show you what the signaling is that turn each of these on. And the ADCC cell is one that has evolved to take care of things when none of the other answers seem to work. ADCC is an abbreviation for antibody-dependent cell-mediated cytotoxicity, and it's a process, as the name implies, that involves cells doing the actual killing, but antibodies doing the targeting of who is going to kill. And so my next step will be to go through the mechanisms by which each of these works. Now, if you'll notice, the cytokines from the T helper 1 that turn each of these cells on are the same. So think of this as being a process of these effector cells coming into an area in response to the signaling from the T helper 1, making the evaluation of whether or not they can effectively destroy the pathogen that's causing the problem. If they can, they are activated and take care of it. If they can't, there will be someone else in this lineage who will be able to do so. Now, there is a special case within this side of the immune response that is the only case where the effector cell and the antigen-presenting cell are actually the same cell. And that is the special case where I have pathogens who are infected themselves inside a mononuclear phagocyte. So if my antigen-presenting cell is indeed the cell that is harboring the invading pathogen, then it would be unwise for me, or at least wasteful, to call in one of my killer cells from down below to manage the problem, because the result then would be that I killed my own antigen-presenting cells. So the mechanism that your body has evolved that is very clever is to try initially to turn on that antigen-presenting cell to see if it can get rid of the pathogen itself. So that particular mechanism is the first one we'll discuss down below. And macrophages, mononuclears, other dendritic cells, and APCs, when they indeed are infected with the material that needs to be disposed of, then the T helper 1 cell will activate those cells to become what we call angry phagocytes, so that they turn on more of their microbicidal mechanisms and do a better job at getting rid of what the problem is. Now, for my monocytes and macrophages, my most distinctive CD marker is CD14, which is the endotoxin receptor. And of course, we'll be talking about that a little bit later on. But this is the preferred mechanism of control for microbes that I refer to as being facultative intracellular. Now, as we talk more in infectious disease, you'll see that what that means is that I can actually grow those microbes in the laboratory perfectly happily on agar or in broth, but inside the human body, they allow themselves to be eaten by these specific phagocytic cells, and they persist inside them by having a variety of different mechanisms to do that. Some of them will inhibit the fusion of the phagosome and the lysosome. Others of them will jump out of the phagosome before it fuses with lysosomes and will live free in the cytoplasm. But regardless, the problem that these are living inside my most important antigen-presenting cells produces a very important problem for your immune response. And so the technique of trying to make these cells do the intracellular killing better themselves is a very good first step to try. Now, of course, we will talk about this more as we get into our discussion of infectious disease, but for now, you should recognize this list of organisms out of the fungal, indeed the parasitic, in fact, and the bacterial groups that live inside mononuclear phagocytes. And so for each of these cases, my most important protective mechanism is going to be to activate my T helper 1 to deliver an interferon gamma signal to the infected cell, which then causes those cells to become highly secretory. We refer to these as epithelioid cells, and the end pathologic lesion, of course, is the granuloma. Now, as a generality, we can refer to all of the responses happening on this side of the nirvana diagram as being granulomatous inflammatory responses. But the specific lesion of the granuloma is one that we name specifically for these particular organisms because it ends up being a spherical accumulation of T helper 1s and infected macrophages trying to collaborate in order to get rid of these facultative intracellular pathogens. My second effector mechanism is the cytotoxic T cell. And as I said, this is probably my most elaborate and specific of my killing mechanisms because the identification of the problem cell is truly exquisitely controlled through recognition of MHC class 1. So this is the best cell to make to control your obligate intracellulars, like viruses, like chlamydia, rickettsia, some of the sporozoan parasites, and so forth, because these are also the most important protective responses against neoplastic change. Cytotoxic T cells, or CTLs, are attracted to the area of an infection or malignant transformation again by the cytokines of that same immunologic general. And as the general instructs them to come in, they are then induced to clone themselves by the signaling from the general. And that cloning causes them to multiply to a very large number of activated killer cells so that they can take care of the problem. What causes them to recognize who needs to die is a signal we spoke about before, and it is the MHC class 1 loading mechanism that I've abbreviated here. Remember that my MHC class 1 molecule is made in the endoplasmic reticulum, and after transport by transporter molecules, peptides made in the cytosol are transported and take their place in the groove of that molecule before being transported to the surface. So if this is a normal cell, it's wearing normal peptides, and there should not be a cytotoxic T cell that would recognize it. But if this cell has a virus or some other malignant process going on inside, then what will happen is that those peptides will be the recognition molecule that will turn on the cytotoxic T cell. So cytotoxic T cells will bind to that MHC peptide combination because of the unique shape of their T cell receptor. They will rearrange their cytoskeleton, and they will then release first perforins that will punch holes in the membrane, and then granzymes that will turn on the extrinsic cascade of apoptosis. Okay? So in a diagrammatic fashion, here is my cytotoxic cell recognizing the abnormal peptides of an infected cell, and then liberating granzymes that will be taken up through the holes in the membrane created by the perforin. So the target cell will undergo apoptosis. But if you notice, there will not be any normal cells in the area that will be harmed. This is a very exquisite and specifically directed response that kills only those cells that need to die. Our next effector on this particular side of nirvana is the natural killer cell, which was actually the first of the innate lymphoid series that was discovered. And because natural killer cells are said to be innate, they do not express any memory, but they're designed to be a gap in between the activation of the innate inflammatory response and the activation of adaptive lymphoid cells on this side of nirvana. Realize that they continue to have a role beyond that, because it's true that many pathogens have achieved a way of avoiding killing by cytotoxic T cells by downregulating the expression of MHC class 1 in the cells that they infect. This is common in many viral infections and also in many malignancies. In the viruses, herpes and HIV both stop the infected cell from expressing that recognition molecule. So remember, in the absence of MHC class 1 expression, that means I cannot be killed by a cytotoxic T cell, and the cell that has to step into the breach is my natural killer. Now, we identify them by their markers. They have CD16, which is the Fc receptor on their surface, and they have CD56 on their surface, which is a metalloendoproteinase, but we actually do not know what it does. That's simply how we identify them in a complex mixture. The target cell identification for killing is a two-system signal on this side, and the receptors which mediate it are in the same immunoglobulin superfamily of genes, so we call them killer, immunoglobulin-like receptors, or KIRs. Now, there has gotten to be a little bit of confusing terminology in the literature because some people apparently did not realize that KIR stands for killer immunoglobulin-like receptor, and they use the abbreviation for something entirely different, which is killer inhibitory receptor. Okay? We are not going to use that terminology. We are going to use the correct syntax because both of the KIRs are not inhibitory. Indeed, one of them delivers an activating signal, and one of them delivers an inhibitory signal. So it's really not that one is a KAR and activates, and one is a KIR and inhibits. They are both KIRs, but one activates and one inhibits. Now, the killing mechanism is the same as we saw with the cytotoxic T cell. It's still by delivery of perforins to punch holes through the membrane and then release of granzymes. But if you notice the difference of the signaling for the on and off signals shown here, the activating signal, the KIR that activates, apparently recognizes a damage-associated molecular pattern on the target cell. There has to be something wrong with the cell. The inhibitory receptor actually recognizes MHC class 1. So in the position where the inhibitory receptor sees class 1, even in the presence of the activating receptor, the NK cell will not kill. But if the presence of damage is reflected in the binding to the activating receptor, and the cell has stopped expressing MHC class 1, then the natural killer cell will deliver the signal that will induce the apoptosis in the target cell. Now, students always ask me at this point, or perhaps sometimes after they've thought about it for a little while and come back, since my erythrocytes are the one cell in my body that does not have MHC class 1 on its surface, why is it that my natural killer cells don't just run wild and kill all of my red blood cells? Well, the answer, of course, is in the two-signal strategy. It's true that my red blood cells don't have any MHC class 1, so that particular pair of receptors is in the appropriate position to cause death. But unless I've got a problem with my red blood cell where DAMPs are being expressed on the surface, I don't have the activating receptor in the appropriate position. So my red cells are not killed because they don't express the damage that is the activating signal for these cells. My final mechanism is antibody-dependent cell-mediated cytotoxicity, which I mentioned before. And as you'll see, we have to get a little bit ahead of ourselves here because of the fact that I've got to tell you that this process requires the product of the other side of the immune response, which is the humoral immune response antibody. So on this side, we're just going to take it for granted for a moment that antibody exists at some times, and if it is present, then the antibody can indeed bind to a cell that has something wrong going on the surface, and the antibody then can be used as the identifier of that cellular damage in order that the cell can then be targeted for release of similar granzymes. So the apoptosis is induced extracellularly, as in the other cases, but the signaling molecule is an antibody molecule that has identified something abnormal on the surface of the cell. Now you'll notice that my types of cells that are capable of doing this are really quite a few. Monocytes, macrophages, natural killer cells, PMNs, even eosinophils. Although in the case of eosinophils, it's an IgE isotype of antibody that's used, and in the rest of the cases, it's IgG. Now again, we haven't talked about antibody as yet, but we will in our next chapter. And suffice it for now to say that it is the antibody that is recognizing the problem cell. The cell is doing the killing, the antibody is the targeting weapon. Now, to think about why it would be true that I need this particular mechanism, I'm going to tell you the story of a particularly unusual virus that belongs to the herpes family, namely cytomegalovirus. If you remember that cytomegalovirus is a herpes, then you should already be thinking about the fact that herpes viruses downregulate MHC class I expression. So as I'm going mentally through my possibilities here, remember my first possibility that would be my cytotoxic cell is not going to work with cytomegalovirus because there's no MHC class I. So the logical thought would be next that my NK cell would be my next option for a cytomegalovirus infected cell. But what CMV does that is terribly clever is it makes a fake version of MHC class I and causes that to be inserted in the membrane of the infected cell. Well, that fake molecule is good enough to fool the natural killer cell, but it's not good enough to be used by the cytotoxic cell. Well, now I've got a problem. I can't use my cytotoxic cell. I can't use my natural killer cell. My macrophages don't work because they only work on facultative intracellulars. So the only thing that keeps all of us from dying of fulminating CMV infections is the fact that an antibody that can be made against the fake MHC class I. It's recognized as a foreign protein, and the antibody against that fake molecule recognizes them on the surface of the infected cell and targets the infected CMV cell for killing. Now, what I hope you're taking out of this is the idea that we have a nearly perfect immune strategy for at least everything that has ever approached mankind on this planet so far. Now, we will be talking about later on the perfect immunological pathogen that I'm sure you already recognize as being HIV. But before HIV arose, there truly was nothing within the panoply of microbial life that was able to avoid recognition and ultimate destruction by this extremely powerful immune response. Now, this is the nirvana diagram, and what I'm going to do here is simply talk through it quite quickly as a summary. You're going to see this again as we go through the humoral immune response. So simply realize I've divided it into two sides, with the humoral being on the right and the cell-mediated being on the left, as has been consistent through these notes. So realize what we did here was that we took our antigen-presenting cell. We convinced that antigen-presenting cell that whatever had caused the damage up above had been caused from within via TLRs 3, 7, 8, and 9, delivery of an interferon gamma signal, and that APC became a classically activated APC. That cell, in turn, communicated to my T helper 0 with interleukin 12 and induced that cell to become a T helper 1, who then became the general who delivered the signaling to all of the effectors of this side of the immune response. My cytotoxic T cell, my natural killer cell, and my ADCC cell. Notice the special case where the cytokines of that T helper 1 stimulate the APC1, that being the classic indication of granuloma formation. And now one more thing I'm going to say to you before we close this chapter and talk about some make me care issues. Realize what I have designed here for your understanding is a diagram that divides in half top and bottom. Please remember that when I am trying to deliver the signal that says help me, that delivery is through class 2 presentation between the APC and the T helper. When I am deciding who is going to die, if there is MHC involved, it's class 1 that allows me to recognize the kill me signal. Okay? So I always require help. The source of the cytokines that turn the entire immune response on is the general who recognizes what excites him, presented in MHC class 2. The effector cells in this chapter recognized who needed to be killed based on either having MHC class 1 or the absence of MHC class 1. Okay? All right. Now, one of the things I might suggest to you is that you take that diagram, because I know it is quite small, take it and print it and have it enlarged. Run some plastic over it and then use it as a technique to work through your understanding of the system. The idea of putting it under lamination is to make it so that you can easily erase it and do it again. But I know people who have enlarged this up to poster board size and use it as artwork in their apartment. And while they're doing that, they're constantly working through the logic of what would be the best thing for your immune response to do in order to solve this problem. Okay, now the hashtag make me care here, number 1, unfortunately, I have just killed. So sadly, I went through that with you, but you can think your way through that again in your spare time. The second one that I haven't actually introduced and killed yet is the MMC number 2, which is in reference to the drug infliximab, which is being used in the therapy of rheumatoid arthritis and Crohn's disease and a number of other chronic inflammatory conditions. So what you should know about infliximab, even though I'm not a pharmacologist, is that it is an inhibitor of TNF-alpha. And if you think about where it is that TNF-alpha is produced in this particular diagram that we just went through, you should be able to predict one of the key concerns we have when we treat patients with infliximab, that they're going to become susceptible to a particular category of infectious agents when you treat them with that drug. Can you think about what category of agents would be particularly problematic and explain why that would be so? I will let you stop your video and think your way through this for a while. And when you come back, when you're ready to come back with your own thought processes, we will work through it. All right, so TNF-alpha, first stop here, okay? The inhibition of TNF-alpha is going to affect the nirvana diagram at the level of the interaction between the APC and the T helper 1. TNF-alpha is one of the secretions of that cell that is going to stimulate my T helper 1s to reciprocally stimulate through interferon gamma. And if you recall, that very specific loop within the nirvana diagram on the cell-mediated side is the one that is involved in the management of the facultative intracellular pathogens. So I always make certain that I know before I put someone on infliximab, I've got to make certain that they do not have a positive PPD test, because any sort of pathogen within the facultative intracellular group is going to be exacerbated in its condition if I were to use this drug in that patient. Okay? All right. I think the only thing remaining is your personal review of the way that you should be able to use these diagrams to think through the process that we'll be talking about. And our next topic will be the other side of the nirvana diagram, where we work through the production of antibodies. Okay? Now, let me put one more thing in your mind before we quit, and that is that even though I am going through these two systems as being very discrete, one against intracellulars and one against extracellulars, realize that it will be true that there are some proteins created at the site of inflammation or entry of an original pathogen that will indeed go down the side toward the production of antibodies. So simply remember that it's very common, even in those pathways that involve cell-mediated effectors to begin with, for some of that material to also be processed and ultimately produce antibodies. And that is indeed how we get the antibodies that we needed for ADCC, as you saw in this chapter.